And so next, it's my pleasure to introduce Alastair Lamb, who's at the University of Oxford and whom I had the pleasure of overlapping with in Cambridge for a brief stint many, many years ago, it feels like. And he is going to talk to us about the spatial landscape of clonal mutations in benign and malignant tissue. Welcome.
Thanks, Christina, and it's a pleasure to see you again. So yes, as Christina says, I'm a clinician scientist. These are my disclosures. And so there are two parts to my job. I cut out prostates. This is our team with our robot in the background. And I also run a small lab with my biologist colleague, Ian Mills, who some of you know, which I'm aware of. And it's a real pleasure to be here in Tampa together, isn't it? I went for a run yesterday morning and I crossed the river to, I think it's called Davis Islands. And I found myself in the middle of the city's main hospital, which has a cancer center right at the heart of it. And at that big building at the back, I think, is the University of South Florida. And in between is the emergency department with its level 1 trauma center and helipads. So a cancer center at the heart of the hospital. And I'm reliably informed by Audrey and Sandy and others that that's not even the main cancer center in town. So you Americans, you do this so well. You find a need, you find the money, and then you build it. It's all about infrastructure, isn't it? Something that I think the Americans lead the way on. And infrastructure is really important in cancer as well. I think because we've largely ignored infrastructure, we actually know very little about the transition from benign to malignant tissue. And that's not helped by the fact that so often, with a few exceptions, Trevor, but so often we bulk sequence this tissue. And so we get really high-level genomic detail, but we lose our spatial resolution. Infrastructure matters. And we're at fault with this in our prostate cancer studies as well. This is an important study which Christina might remember from my former lab in Cambridge, the complex men study, where we do take several samples from the prostate, but we then bulk sequence them. We were able to derive, I say we, I wasn't actually on this paper, my boss's paper, derive clonal phylogenetics, but it was largely guesswork filling in the gaps in between. So this is where we've been in prostate cancer. So it's really important to fill in the infrastructure. And I think if we do that, we might finally end up discovering the information we need about that transition from benign to malignant tumor. And let's face it, with the exception of Inigo's wonderful talk on Tuesday, an entire talk about benign tissue, we largely concentrate on the cancer, or at least what we think is the cancer, don't we? So our objective in this work was to study spatial genomic integrity in a spatially resolved setting to derive clonal relationships. And we do this using spatial transcriptomics. So I hope that most people here in the audience are now familiar with Visium. It's been available commercially for about 18 months and is based on a technology from Joachim Lindberg and others' group in SciLife Lab, Patrick Stahl's paper back in 2016, sorry. And initially, we tried to use transcripts alone to derive clonal hierarchies and met some problems with that. So we decided instead to turn to copy number. And we used an adapted method from another science paper, Anu Patel from the Broad Institute, to infer CNVs, and we did this in a spatial way. So we have 3 layers here: inferred genome, observed transcriptome, and this is underwritten by really detailed pathology. In this study, there are over 150,000 spots that we've analyzed across a number of tissues, although I'll be focusing on prostate mainly here. And Sam, your colleague, Thomas, has been hard at work here, annotating each of these spots to derive a consensus pathology call. And this is so important for the work that I'm about to show you. But in recognition that even those who might accept that copy number-based clonal phylogenetics is okay, you may still want to know that our inference technique is correct. And so we turned to what is generally accepted as a monoclonal tumor. This is a pediatric brain tumor. And just very briefly, on the right, that heat map, each row of that heat map is a spot. So there are about 4,000 spots on this. And in this setting, it is monoclonal, and hence that sort of red clone labeled A on the right is just a single color overlaid on the histology map on the left. And because it's monoclonal, we can use bulk sequencing to validate this, which we did, and you'll be pleased to hear that the chromosome 3 gain and the chromosome 9 loss and gains were recapitulated with whole genome sequencing. So back to the prostate, and this is a whole organ analysis now. And so we take a 5 millimeter axial section, and rather than those little sort of watch battery buttons, those 5 millimeter circles in that Nature Genetics paper, we now cut this into cubes and take a single section when we get down to, we cut through to cancer from each of those for spatial transcriptomics with that 3 layers that I've talked about, inferred genome, observed transcriptome, and detailed consensus pathology. And we use this to assign each spot to a particular clone family and then derive phylogenetic trees. So let's look at that section. This is it. It is 2-dimensional in the sense that it's an axial section taken from the prostate, but it's 3-dimensional if you accept that there's 5 millimeters of it. And we can observe this as a sort of schematic. And you'll see straight away there are 2 sites of cancer, but there's lots of benign in there, and that's really important. And remember, across the entire section here, we've probably got 80,000 spots. Compare that to the 5 that we were looking at in the complex men study. So let's look under the hood here, as you say in America. What's going on? So here's one section from the right-hand side, the big tumor focus. And you see now the spots overlying that. And this is the histology calls with red being cancer, blue being benign, and a sort of transition state in between, which we won't go into today. What are we actually looking at here? Well, this is what Thomas has been doing, Sam. So you know, really every single spot, which incidentally represents about 10 to 15 cells, has a consensus pathology call, and we only permit that call if there's more than 50% of a particular cell type. So where that stromal layer weaves through the middle, that's the black areas, we exclude those. So I mentioned that there were many, many benign regions in this section, 60,000 spots indeed, and so we looked at them first. And interestingly, while we expected, the reason for doing this is with this spatial inferred CMV approach, you do have to set a reference set, which is your sort of diploid group. Nonetheless, you can see in this map, while there is diploidy for most of it, there's an area which clearly has some copy number events. Interestingly, those that we might normally associate with prostate cancer, so chromosome 8p loss and 8q gain. So we were sort of referring to those as our altered benign, and we selected another region which would be our sort of pure benign reference set as we go forwards. So we wanted to look at the section which had the most of those altered benign spots in, and it was actually this one down here on the left, the smaller region of cancer, clinically significant cancer, but still smaller nonetheless. And here's a sort of map now at the spot level of the histology. Blue is benign, and red is cancer, with a couple of high-grade cancer areas here and intermediate risk here. So what we can then do is look at that heat map. Now, this is equivalent to what we saw before, but this is now this one section. So this is about 1,000 spots. We've excluded all non-epithelial for the sake of this analysis, and we've excluded those that are less than 50%. And you can see when you get down to this granular level, it's actually really clean. So you can assign clones quite easily. And when you look at those overlaying on the histology map, you can see they do follow the branching morphogenetic pattern. This is how prostate cancer develops. We know this. And those clones follow the pattern. I don't have a sort of more zoom in, but you can do that, and you can see branches coming down from the top here, working their way through the yellow into these clones here. Now, this is really important. If I just go back one, you see that these clones traversed histological boundaries. Let's focus particularly on the yellow clone, which contains many of the features which we would think of as being pathognomonic of prostate cancer, are present in this purely benign region. And the other clones traverse benign and cancer. And we think this is really important, potentially the first time that this has been observed. And of course, when you have this level of granularity, it's not difficult to construct a clone tree. And I've made this case a few times to a few folks in discussion over posters, that kind of thing. I think this clone tree, you can kind of maybe take my word for it, but I can show you in more detail if you'd like, does basically follow the branching morphogenetic pattern of this region of glandular prostate cancer tissue. So, for the sake of time, I'm going to skip through these next few slides because I would love to leave some time for questions, but we have validated this. We accept that some people may still not believe us, the reviewers have asked for a number of validations. We've done this already before we sent it in, with fluorescence in situ hybridization, looking at 2 key prostate-related oncogenes or tumor suppressor genes, MYC, PTEN, and we confirm what we expect from the inference with the validation. We've also done the same with inferred SNVs and variant allele fractions for the small region of the exome that we have access to. And we've also constructed an in silico model where we basically seed fictitious clones, create a grand truth, and then validate that with our spatial inferred CNV method. So what's next? Well, if you accept my argument that we now have a tool to properly characterize in situ the infrastructure as well as the cancer itself at a spatially resolved clonal level, then this may finally allow us to create that link between the lethal clone in situ and, as Charlie Swanton was saying yesterday, the interplay between that and early micrometastatic seeding, something which is still really poorly understood. What else did Charlie say yesterday? Well, what about the tumor microenvironment? What about the interplay between the stromal niche or perhaps clone-clone crosstalk? And early detection and prognostication. When we biopsy, are we biopsying the lethal clone? Have we found it? Do we know which is the lethal clone? Or when we radically treat, do we know if that clone has already escaped at the point of radical treatment? If so, that could inform adjuvant therapy or potentially even neoadjuvant therapy. These are all really important questions, but I think we now have some of the tools to potentially answer them. So acknowledgements are to Joachim Lindeberg's group. They invented this spatial transcriptomic technique, and it's been a real pleasure, particularly during COVID, where we lost access to some of our wet lab to be able to access some of their material and work together on this project. And I'd like to acknowledge as well my team and collaborators and colleagues in Oxford, or not in Oxford. But thank you very much, and I'd love to hear your thoughts.
Thanks, Alastair. All right, kick it off.
Great talk. If I understand correctly, in the prostate, the lesions has been found out quite different and heterogeneous. So, have you been able to take a look at those in the heterogeneity inside one lesion and compare to the other lesions? So, how much they actually differ. And the second one is that there's also, if I'm correct, there's a kind of cross-seeding. So, do you see that there also in the prostates?
So I've only, because of time, been able to show you one sort of our vignette stories. There are many. Do have a look at the pre-printed paper, and hopefully we've just sent it back to a well-known journal when it comes out. I'm very interested by your comment, Sam, about we know there's intraprostatic seeding. I'm not sure we know that. It may happen in other organs. We have looked at that other big cancer focus over on the right, and we've seen 2 or 3 different clones. Interestingly there, what looks like, we think what we see is that the branching morphogenetic pattern then stops, and you get that, remember that big red clone, that big red outgrowth? Well, you saw red histology. It is also a big red clone, and it's basically pushed apart the branches at that point, because that's a high-grade clone, it's proliferating. Your question about seeding is fascinating. One of the reviewers said, why have you separated your phylogenetic trees on the right and left side of the prostate? Isn't there evidence for a common ancestor? That's really controversial. Can a seed metastasize within the prostate? Conceptually, it might. There are little lymphatic channels that run through the prostate. There are blood vessels. I don't think we quite have the evidence yet to be certain of that, but we'd like to push down that route a bit further. Great questions. Thank you.
Carlo.
Yeah, a really wonderful talk and a great new view and detail of the evolution going on there. I noticed in your plots that the clones were generally contiguous, but there was also little scatterings of clone fairly far away amidst the other clones. Do you have an idea of why that is? Is that a mistake, or is that something about the 3D architecture of things, or is there micrometastases? What is that?
So, yeah, love the question. So we are sort of looking at 2 dimensions here, aren't we? So these are branching glandular structures which will be going in and out behind. So where there's scattering, there may be a 3rd dimension which is contiguous. So I think that's important, if that's what you're referring to. If you're referring to broader scattering, I refer you to my answer to the previous question. It may be true, there may be skipping events. but that's controversial. Let's see if that's really true. What was your other question? Sorry.
Oh, it's just that one question. I was wondering what there's different hypotheses that might explain why you would have a little focus of one clone in the midst of another clone. And as you say, one is that there's complex three-dimensional architecture. It's all contiguous, but we see something similar in Barrett's esophagus, which is two-dimensional, and we're trying to understand why you would get like a minority clone showing up in multiple places.
Yeah.
In Barrett's esophagus, anyway.
So I guess I'll give you a boring answer to the question. Inherent in our spatial and first CMV approach, we require spots to have at least 10 spots neighboring each other to be defined as a clone. So there need to be at least 10 spots, and they need to be next to each other in order to, and that is why it's very reproducible, we hope, but it's why that in first CMV heat map does look quite clean. So we do try and remove some noise. But in doing that, we may actually have lost some biology, which could be meaningful. And I think it would be interesting to push further down that route as well. Thanks. Yeah.
All right, one last quick one. Jake.
Hey, Jake.
Hey, that was beautiful. So, you and I differ in that you like to take organs out and I like to leave organs in. Can you imagine?
That can cause problems, they can't do.
Well, it can do, yeah. Well, so can taking them out.
That's true.
And in prostate cancer, you take the whole thing.
Yes.
But maybe you don't have to, right? So
Right.
I'm wondering, I clearly see the value of finding the thing that in the prostate which kills you genetically. So you take it out, and then you figure out what's going to kill the person, and then you would have something then to go look for in their blood or something, right?
Yeah.
But is there a way we can think about your method in a way that doesn't involve removing the whole thing,
Yeah.
and therefore can help us to understand focal sub?
Yeah. So, Jake, the final line of our abstract is this may inform focal therapy in the future. I don't know where you think you stand on the focal therapy paradigm. I'm a bit of a disbeliever.
I don't believe in HIFU, that's for sure.
Right, okay, so neither do I. But not because I don't think it could be useful in the future, but because I don't think we understand enough about the biology of this heterogeneous multifocal disease to be treating it focally, where two things. One, we haven't actually properly sampled the prostate. MRI is great, but it's not perfect. So we don't know about small contralateral lesions, which may be the dominant lethal lesion. Also, as we've shown, we need to drill down into quite what the difference is with these clones that traverse histological boundaries. But let's say that yellow clone is actually pre-lethal. Well, it's not cancer, and based on our sort of understanding of how focal therapy decisions are made, it may not be thought necessary to include that in the focal region of interest. So I think this kind of info, I hope, will inform focal therapy. I'm not a disbeliever in the future. I just think currently we're trying to do it when we don't understand the biology of the disease.
Agreed. Thanks.
Great. Thanks again, Alastair. Really beautiful work. So now, if